Thank you, sir. First of all, I would like to thank Scientific Committee AIOS and also Dr. Dinesh Garg and Dr. Arvinder Gupta have been very kind, and we thought that we could schedule this after the ARMD because it forms relevant. So I'm here today going to speak on the evolving Ranibizumab Biosimilars, a developmental and a clinical review, the story of Razumab. I have no financial disclosures in this product. As we all know, biologics are highly complex molecules. The monoclonal antibody forms around 20,000 atoms. So how are these defined? The biologics are nothing but a wide range of products such as vaccines, blood and blood components, and recombinant therapeutic proteins that prevent, treat, or cure a disease. The reference biologics are nothing but the originally licensed biologic product used for comparison, whereas the biosimilars is highly similar to the reference product with no clinically meaningful differences in terms of safety, purity, and potency. The biosimilar development in India is done through the guidelines on similar biologics by the Department of Biotechnology and Central Drug Standard Control Organization, Ministry of Health and Family Welfare. So in Indian perspective, similar biologics, that is the biosimilar product, are developed through sequential process to demonstrate the similarity by extensive characterization studies revealing the molecular and quality attributes with regard to the reference product. So why do we need biosimilar ranibizumab? There's a lot of unmet clinical need. Like if you see in the Indian population, there's more than 1 crore of people being affected by either the wet ARMD or the diabetic retinopathy, macular edema. Indigenous biosimilar introduction in developing country like India will help reduce substantial cost burden. The risk-benefit assessment was quite favorable. If you see the considerable evidence for safety and effectiveness, it has higher compliance and broader therapeutic applicability. So this recombinant ranibizumab, as we all know, it's an antibody Fab fragment targeted against human VEGF-A, which has a light chain and a heavy chain linked by a disulfide bridge. So I'll not go into the details of these clone, but to just touch a brief, this is the light chain of the ranibizumab and the heavy chain. The gene of interest is taken and put it up into the vector. And then it is introduced into the host cells where it multiplies and it produces the ranibizumab protein. So biosimilarity assessment, if you see, they go through a lot of tests like the comparative force degradation studies, that high pH or high temperature, forced oxidation. They go through the purity analysis like process and product related impurities, HPCL, electrophoresis, mass spectrography, etc. The structure, the primary structure, secondary, tertiary conformation, and of course the bioassay and the binding kinetics. So the peptide mapping with the innovator product was done. If you see the up and the lower, it was quite matching. The secondary and the tertiary structure, the primary, secondary, and tertiary structure of Intas ranibizumab was quite matching with the innovator product. There are actually 2 lines here. One is the green and one is blue, which shows that they are quite matching. The purity of these Intas ranibizumab molecule was also tested in the form of cationic exchange and in the lipid chromatography, and it was found that both the products were pretty much similar. If you take into the potencies, the human umbilical vascular endothelial cell line, which is very rich in the VEGF, that was taken and studied the binding kinetics and the potency of this ranibizumab molecule with that of the reference product, and it was quite matching. A lot of preclinical toxicity studies were done on the Wistar rats, Swiss albino mice, New Zealand white rabbits, guinea pigs, etc. The immunogenicity analysis, all the analyzed samples were found to be negative for anti-ranibizumab antibodies. Then what the hell happened? July 15, 2 cases of intraocular inflammation reported at Surat. Cases resolved in 3 to 5 days with topical and systemic steroids. Additional cases reported from multiple other centers. And in our center, 12 cases reported with mild degree of AC reaction, which resolved within a week's time. So what might be the reason for the inflammation? There might be either, is there any denaturation of the drug, if the cold chain is not adequately maintained, or whether there is any problem in the sterile inflammation due to varied OT practices or practice patterns giving the injections, or whether there was any reaction to the drug or the excipients. Then when I asked the R&D of the Intas Pharmaceuticals as to how it happened, then I was explained about how they are trying to solve this problem. So in the processing of this ranibizumab molecule, they added the additional gel filter to the bulk. And if you see this formulation, because it was a tough, challenging task for them to find out how did this reaction were happening. So they made 4 different arms where they studied. The one arm had ranibizumab along with all these excipients. The second arm had ranibizumab protein alone. The third arm had histidine, hydrochloride, and trehalose. And the fourth arm had all of these. And then they found out the arm which had the polysorbate 20, that was showing reaction. Then they changed the vendor to different vendor. And after that, the fresh ultra polysorbate was obtained, and then it was distributed in the market, that is drug 05 and 06 batches. Then we conducted an observational study, a pilot multicentric open label study to observe the acute immunological reaction to this razumab molecule. The primary objective of this study was to observe AC and vitreous for any evidence of inflammation post-administration of razumab. The observation schedules were done on the baseline, day 1, day 2, day 7, and 1 month. Although there are more than 1,000 cases of razumab being injected in India, but we have taken only the cases who have complied with all the protocol. That is, who have followed on the baseline 1, 2, 7 days, and 1 month. So we could collect the data of 213 patients till now. And this is how it was divided. Nearly 40% of the patients in this razumab study group were around in the CNVM, and 34% of the patients were with diabetic macular edema, and 27% of the patients suffering from vein occlusion. And if you see this graph, not even a single patient had any ocular inflammation. The mean change in BCVA, yes, there was some mean change. If you see from 0.71 logMAR to nearly 0.53, but this is varied of all together, all different diseases together. If you see diseases in particular, if you see CNVM, you had around 0.8 to 0.6. So there was, yeah, good visual recovery. And if you see the DME from 0.6 to 0.4, and in retinal vein occlusion, it is from 0.7 to 0.46. The mean change in OCT overall, inclusive of all diseases, if you take an average of 401, then at 1 month it was around 306. So in CNVM, we did see some decrease in CMT. DME, yes, from 392, there was some improvement up to 300 microns. And in vein occlusions, we did see substantial improvement from 434 to 293. So I'm just going to share some of the clinical photographs and end my talk. So this was a patient presented to our hospital with juxtafoveal CNVM, 70-year-old male. And if you see, there are few cystoid spaces, intraretinal cystoid spaces. This is a day 1 picture, day 7 and 1 month. There was some reduction in the CMT and disappearance of the cystoid spaces. And this was another patient who came, with 63-year-old male with a diabetic macular edema. If you see, there is a central cystoid space, foveal edema, and at the end of 1 month, there was reduction in the central macular thickness. This was a patient with a BRVO macular edema who had more than 600 microns of macular edema, but after 1 month, he did show remarkable improvement. This is a case of CRVO macular edema, 43-year-old male, who again had a CMT of nearly around roughly between 600 to 700 micron, and there was some improvement on day 1, and day 1 month, we see there is complete resolution of subretinal fluid and the intraretinal fluid. So this was another patient of CNVM. We see some cystoid spaces here and the subretinal fibrosis down here. But as we go on to the 1 month, there was reduction in the central macular thickness, or the CFT, what we call central foveal thickness, and disappearance of the intraretinal fluid. And see the DME, diabetic macular edema, the patient had SRF, foveal as well as non-foveal edema, but at 1 month, there was complete resolution of subretinal and intraretinal fluid. And this was recently just taken 2 days back. There's a patient who came to our hospital, and because before meeting me, they already come with the OCTs, and the patient the patient himself said, "Doctor, I don't need injections." I asked, what happened? Then they showed me both the comparative figures, and because, as you all know, the patients have now become very smarter because they try to compare the OCT previously and today. And then they ask, where is the edema? Oh, doctor, there's some fluid. I need injection today. So that patient was quite bright enough, and he said, I don't require. When I asked why, yes, there was a subretinal fluid, and there was completely disappeared at the end of 1 month, and he walked away happily. And this was another case of diabetic macular edema. If you see the large foveal cystoid space, had very good resolution after 1 shot of Razumab. Another few cases. So we're looking for a way forward, although we don't have comparative studies as of now. So we are trying to have a multicentric randomized control trial for comparing Razumab with the innovator product in vein occlusions. So I just discussed it in the open podium. If anybody interested to participate in this trial can please either telephone or mail to this email ID under Dr. Guru Prasad. So we are trying to head it and see how it goes between head-on. So in conclusion, Razumab or Intas developed a high matching and stable ranibizumab with desirable quality attributes in India. The fresh polysorbate 20 and the filtration was further added to process to strengthen the quality. The pilot study in 213 patients as of now shows no evidence of acute immunological reaction and safety concern. Thank you so much.